we take an approach which is hypothesis free. What does that mean? We believe in data first. So I would define what I would do. I would define the question, I would set up the question, and I would show you that how, when you start with the data without any hypothesis, where to look at, and then how this data, if you could statistically and a significant in a principled manner, if you could really pass that data, then how you can derive a biological sense to it, and that would actually then go back to the clinics to benefit the patients. Now that's the strength of the Tata Memorial Center, where I kind of come from. So I'm going to specifically restrict my talk to the lymph node metastasis in tongue cancer. But before I get into the tongue cancer, let's get a complete whole overview of the landscape, what exactly is happening in India in just a 1 minute. This is my favorite slide. It's an old slide and I always keep in my presentations. What it does is that it is a snapshot of 1950 to 2001 of mortality across different diseases. And what you would see that whether it is a cardiovascular disease or it is a cerebrovascular disease or an infectious disease, over this time period of 1950 to 2001, there has been a significant decline in the mortality. And why did this happen? This happened primarily because advancement of the underlying biology, whether it is the cell wall synthesis, whether it is cholesterol biology, you name it. When it comes down to cancer, unfortunately, there wasn't any drop, and the bad story is, the sad story is, that now, and that's the reason this slide is old, now you slide this time window from 2001 to 2022, it is now creating barricades between the countries with a higher income group and the countries with a lower income group. If you would look at the data that comes around from countries with a lower or middle income groups such as India, this remains just the same. So that's where the stand my laboratory has kind of taken up to do something which is prevalent to characterize the cancer type which is afflicting the among here in the Indian population. This is what is being projected by the World Health Organization. This is how the cancer burden is going to change and going to increase over the times. So as what you would see in the next few years, this is how it's going to be the growth. Now this growth in the incidence and the increase in the cancer is primarily going to be contributed by, as what you would see by the countries with the middle or the lower income group. And here, this is an old slide. I think this is from 2019, and that's why it kind of shows that there is a trend of a plateau, but not yet a plateau. In the next slides, I will show you that how the countries are getting demarcated. This was the first report in 2019 that was reported in the US, where, much similar to the slide that I showed earlier, a significant drop in the mortality, even in cancer, but in the US. This continues, this goes on to 2020 and on. And now there are more and more countries in the higher income group that is reporting it, but unfortunately in the Indian settings this is way far of fantasy too. In fact, I would show you something more dreadful. This is the data from the Tata Memorial Center about the patients who are walking in, and this gives you a kind of an overview that how is the incidence and how is the disease of, and why or why is the mortality not really decreasing in India the way you are trying to get to see in the Western nations. And that is primarily because the disease in India is also very distinct because of its etiological agent, because of its ethnicity, you name it, we really do not know. So if you would see, another drastic thing is that even though we have less cancer incidence in India, if you would compare the cancer incidence in India as to the Western world, primarily to the Caucasian population, to the US primarily, in 100,000, if you would look at the data among men, there would be 110 to there would be 310 to 320 cases in men, and about in women, and about 300 odd or 290 odd, and among men in every 100,000. When it comes down to the Indian population, this would be about 110 among women and about 94 in men. So absolute number of ours is still low. That is correct, that's a true fact. However, we tend to die more when we get the disease here in India. That is shown around over here. The cancer incidence in India over this time frame has increased from 0.5 to 1 million. However, if you would look at the trend in the increase in the death, it is 0.3 to 0.8 million. So when we get cancer, we tend to die more as you compare to the Western world. So what my laboratory is doing, my laboratory has been working in the cancer types which are like most prevalent in the Indian settings. We are primarily working on, if you would look at the single side, lung cancer still remains one of the most common among men, and that is true across the globe, and that is also true among India. However, lung cancer is a single site disease. But there is another group of cancers of multiple sites which is tobacco associated and which is called as head and neck cancer, and where there is a combination of different sites. If you would add them all together, that is the one which is the most cancer, that has the highest incidence and affecting the Indian population. And for example, if you would just add the lip and oral and you would add the pharynx over here, that is much higher. So the cancer type which is most common in India is the head and neck cancer. Now head and neck cancer, which is so high in India, is not even a major disease in the Western world. It is about 6th or 7th or 8th of an incidence. Similarly, even though breast cancer now is the most common among women, the second most is the cervical, which till few years back was the most common among women in India. Cervical cancer does not even figure in the top 10 in the US. Why are people going to work on it? Why are people going to, why would that much be an attention on an oral cancer? So, gallbladder cancer, we are sitting in Delhi, it's one of the hot seats across the whole globe. You would say that Delhi is one of the very hotbed and among the North Indian regions by the Ganges. Maximum incidence across the globe that you get to see around over there. But it's a rare disease, would you believe that? It's a rare disease in the Western world. Why would anyone work on that? So the initiative to understand the complexities and the nuances such that it could be brought to therapy has to happen around from this region of the world, and that's what my laboratory has been kind of like working. However, today I'm not going to talk you into like any more, I would restrict myself to just one side. I'm going to talk about is tongue cancer. And even in tongue cancer, I'm going to talk about today of a very specific problem. This is an important slide. I will take about at least 40 seconds to take you through what exactly is the problem that what we are addressing, and then I will show you through the next slide that how we are, what we are getting out from the genomics. So one of the major problems in the tongue cancer is a lymph node metastasis. What happens is that if you diagnose at an early stage and diagnose at a late stage, if you diagnose at a late stage, there is absolutely no options much left. There is only surgical options, chemoradiation, and the survival is only 20% for 5 years. Now, that is not very surprising because it's a late stage. But it's surprising that still, it's an oral cancer, right? You can just kind of like walk up, you can ask the patient to open their mouth and you can see it. Why does it really reach to this late stage? All the dentists in India and all other places, they should be able to present it and they should be able to report it at an early stage, but it does not happen. It's a very specialized field and people really are not able to report it even at an early stage. So this is what is the raw truth. Another complexity that what happens, even if you report it at an early stage, what happens is, our body has about like somewhere around 700 to 800 lymph nodes, okay? Of this 700 to 800 lymph nodes, around 200 to 300 lymph nodes are out there in the head and neck region. So this region is very rich in lymph nodes. So what you tend to get to see is a lymph node metastasis, and that too an occult lymph node metastasis, that is what is called. That is a hidden lymph node metastasis. So what happens is that in that early late stage, 70% guys and folks are fine. They do not have any metastasis, but 30% individuals, they got some hidden lymph node metastasis. Now what does a hidden lymph node metastasis mean? They have a distinct survival as well. If you do not have a lymph node metastasis, 80% chance of surviving for 5 years. If you do have, 40% chance. Now, what is a lymph node metastasis, which is so hidden about it, is that the current technology cannot detect it, cannot diagnose it. Whether you do it by CT, MRI, PET CT, you may have heard about it. These are the standard ways how you would diagnose. It goes undetected. So because it goes undetected, these guys cannot be treated. Now there were several different clinical trials that were performed, and they finally realized that the easiest way to deal with that is to completely, is my time up? 5 minutes. 5 minutes, okay. Okay, so I'll do it in 5 minutes. In the lymph node metastasis, whenever it's there, the only option is to resect and do a head and neck surgery. that is to remove all the lymph nodes along with the tumor, assuming that even these guys who do not have lymph node metastasis, but there is no way to distinguish them, they undergo this surgery where there is a lot of morbidity associated with that, or a lot of cost as well. Now, to spare these 70 guys, the molecular biologist can come forward. Molecular biologist can kind of like look for reliable molecular markers which can help clinicians to spare these 70% guys who does not need surgery. So we set into it, and then that's what I would show, that what we have done. I will walk you through. These are not details. We took about our first study. We took about 58 odd samples with comparable number of lymph node negative and positive samples. We went ahead and we performed an whole exome sequencing. This is one of the first data. Details are not important in this slide. This is only to show you that this is one of the landscape of like alterations in the whole exome. Of these node positive and node negative, and based on these, because we did not know where to look at, based on this we find that okay, those mutations that you would otherwise expect to find in head and neck cancer based on the Caucasian population, yes, we do find. So those are fine. So let's go and make some discovery. So what I'm going to talk from here on is that now there is one genomic data, and of course we did more, but it's again the genomics data. From this genomic data, what all information can you really derive, and how that can drive the biology? So one of the first discovery that what we made is that from this data, we found mutations in a gene called as Notch1. And Notch1 that we found that could also be detected in the clinics by IHC. I'm not taking you into the details today. What we went to show that these Notch1 mutations were activating mutations, and that was totally not reported the time when we reported this, because Notch1 was known to be in head and neck cancer as a tumor suppressor. We found activating alterations in Notch1, and that led to therapeutic intervention. A clinical trial is getting initiated at Tata Memorial, because these patients, and what we showed is that patients who are chewing tobacco and present themselves at an early stage, and they are positive for Notch1 mutations, are likely to have more metastasis. There you go. The clinical trial is going to validate soon. Next, we looked at the structural alteration, and we found that there was an amplification of another gene called as matrix metalloprotease 10. Not taking you into the details, I just want to show you that when you do your statistical analysis has to be of extreme high rigor, so that you will be able to find a driver event, because the nuance in the cancer genome is huge. We find Notch1, then we find MMP10 to be amplified. Again, we do a series of genetic alterations, and what we do is that something of the strength is that my laboratory has a strong strength of like having an attack track of an animal house, and we go into an orthotopic mouse model. What I'm going to show over here is a small movie. I hope you got your popcorns along. If not, I'm sure the organizers will be happy to provide you in this movie. What we are doing is that we make the tumors right where it matters the most, right into that organ. What is being done is this is an anesthetized mice, and in that anesthetized mice, the tongue is being extended, and in this tongue we are injecting the cells where we have done all kind of genetic manipulations, whether it is a knockdown of MMP10 or if it is an overexpression of MMP10, and then in some time what you see that this bulge is going to diffuse, and soon you would find that this is how the mice is going to look for, and exactly in an invariant way in 24 to 25 days what we found that there are tumors in the mouse. Now what does that mean? We have an orthotopic mouse model to ask questions of lymph node metastasis. Now these guys, these cells have a luciferase tag in them. Okay, so you don't have to kill the animal. You can do a whole body imaging of that iris imaging of those, and we can see that whether the metastases are happening or not. My laboratory has been doing that for the lung cancer. We have established very elegant models also for the lung cancer without even kind of like cutting open the mice. We have shown that that has clinical relevance. We have very elegant, the only models that I know from here in India, orthotopic in lung or in cervical cancer. Now we have also developed into the breast cancer. However, that is not the part of it. So we went ahead with the MMP10 using an orthotopic model. We kind of like showed that MMP10 indeed, now again this is going to the clinics, MMP10 again is a marker that will allow the clinicians, this paper has been recently kind of like accepted, this would allow the clinicians to make a distinction between who should go to surgery and who should not go to surgery. and we had also dissected the whole, a huge amount of work to the whole pathway by a graduate student. My laboratory is very much interested in other aspects of it. The reads that do not align to the human genome. We are doing, remember, we are starting with the genome data. There is a good amount of data that aligns to the human genome, and that is the exome analysis and all other things that we do for the coding region mutations. But there are a huge amount of sequences that do not align to the human genome. What are those? We developed our own tool, infectious pathogen detector is what we kind of like did, and we went ahead and we looked for the pathogens that were associated. And lo and behold, we made a discovery over there. We found that there is a pathogen, Fusobacterium, which is known to be associated, it's an oncobacterium, it's known to be associated in colorectal cancer. We found a strong association in tongue cancer. And so right now we are kind of like going ahead with the functional work of that, and we showed all the different clinical correlations of it. So now that is also kind of like published. So what I'm trying to show in this is that basically from the genomic data you find mutations, you find copy number alterations, you find pathogens, and more recently we made a major discovery in the field. We have found a novel fusion event. The fusion events, such as you may have read in your textbook as BCR-ABL, and now in the lung cancers as well, such as ALK, which is transforming, we found the first fusion in head and neck cancer. This was only to show that there is a lot of statistical rigor that goes into and to prioritize the candidate, and this is what all the genome biologists do to confuse people. They just show the surface plot, and absolutely it means nothing. You know, I mean, what sense can you make out of this data? Come on. So I'm not going to take you through the data. I just wanted to show you that we found the spanning reads. We made this discovery. We went over, there's a humongous effort that got through a Bhaskar Dharawat, the graduate student, very talented graduate student, went all the way to the whole genome sequencing and find the breakpoints even at the DNA level. And we found a lot of like reciprocal complex events. I will not take you through this. I wanted to show you that was a mismanagement in my head. I wanted to show you that these fusion are activating, and not only activating, it is transforming. When you bring it to the cells, it allows to form tumors, not only in under the in vitro settings, also in the mice. It forms tumors, if you would see over here. Bingo. What comes now is that there is already an FDA-approved treatment. Earlier today, Dr. Amit Singh has already shown over the repurposing of the drug. Here there is an FDA-approved anti-helminthic compound, which is pyridium panoate. And this is already approved. It is being given to the patients. We show that this guy, when given under in vitro conditions, that kind of like, because our fusion event activates the Wnt signaling. This significantly inactivates the beta-catenin, and we show that in the cell line, and then we are absolutely excited that we could really, when we validate this in the in vivo settings, this is an unpublished, we still have to communicate. I really wanted to share with you something which is not from the lab and unpublished. We could show that this is a valid treatment. We are hoping that once it is done, we would be able to do a clinical trial as well. That 6% of tongue cancer patients are harboring these fusion events, and those we found a remarkable response in the animals using these models. So what does this mean? That you may have seen this in the textbooks now, that if you do one NGS sequence, then in that one NGS sequence you can find mutation, you can find deletions and amplifications, you can find translocations, and you can find. So this is what the textbook says. And what today I have done? Today I have presented exactly the same thing: NOTCH1 mutation, MMP10 amplification, fusion of like ubiquitin LRP5, and then the Fusobacterium using this. So that's what we do. So that kind of like brings me to summarize my work. Genomic characterization, that's what I kind of like presented today. I talked about briefly to the notch pathway. I talked about the whole MMP10 signaling pathway that we have dissected, just an overview of that. I talked about the Fusobacterium discovery in head and neck cancer, in the tongue cancer in particular, and about the discovery of a novel fusion transcript not yet published. This is a lab. Here are my functional team members. That does not mean these are non-functional, just that these guys are computational guys, okay? There is a big computational team around over here as well. They will kill me if I call them non-functional. And the clinicians, being at Tata Memorial, we work with the clinicians day in and day out in different cancers, very generously funded by the Tata Memorial Center, by the Department of Biotechnology, Wellcome Trust DBT, DBTV NCI, SERV DST, and the Terry Fox Foundation. So that's, I bring my end to it. I really want to thank the organizers, and I really want to kind of like say it out that the ICGEB has really cut all of us, all these alumni, real deep, and it has really impacted each one of us. This affiliation that has transformed the days that we spent as a graduate student here are the prize moment that we shall cherish forever. Thank you for listening to this far. Sorry for excellent.
Excellent, Amit. Excellent. I mean, you overshoot that time, but the early part where you talked about the nostalgia was fantastic. Of course, the science was also great. So, so any question?
Sorry, couple of quick questions. Great
Sure.
talk, really nice. The genomic profile, I noticed you were mentioning specifically HPV negative. So in a HPV positive setting, presumably, it's quite different.
Yeah. So we earlier published, that was in British Journal of Cancer, we showed that Indian tongue cancer patients are very distinct, that they are all HPV negative. Oral cancer, there are like 30%, and we have shown now that those folks who are HPV negative are actually positive for fusobacterium. And in the TCGA data now we show that in the TCGA in the Caucasian population, those folks who are, these are mutually exclusive sets. Those folks who are HPV positive are not fuso-positive, and those folks who are fuso-positive are not HPV positive. They are mutual subclass with different subtypes.
Okay, that's nice. And the fusion protein, does it retain a ubiquitin ligase activity?
So there is a reciprocal translocation. I could not tell you into the details. So it is more driven by the LRP5. We have done the reciprocal as well with the ubiquitin kind of thing. That is not driving, but the LRP5 where the Wnt signaling gets activated. That's how we kind of like that.
Sanjay, question.
Yeah. Hi, Amit. This is nice work.
Thank you.
So now I know you are the PhD student, so I will call you back to my office.
Anytime. I look forward to that.
One of the questions I have that we are watching head and neck cancer very closely, and you know that we are in the area of neoantigens. So is head and neck cancer going up in India as compared to the Western world because we have not immunized our population for HPV?
So, answer to that, is that HPV vaccination bringing down the incidence of even cervical cancer is not like that very clear, even in the Western population, because there are several other different variants to it. But I'm a great proponent of HPV vaccination, and any day that I would kind of like propose anyone to go for. Whether the HPV vaccination is also impacting the incidence of head and neck cancer, I am not aware of if that data exists around at all, even in the Caucasian world, even in the Caucasian population. As it comes down to the head and neck cancer, into the oral cancer, now we were one of the early ones to kind of like show that, but now it is one of the well-cited paper from my lab. It was in the British Journal of Cancer, and several citations to it, I believe more than 17 odd, and people have reiterated, and now it is very well clear that HPV infection in the Indian oral cancer, primarily the tongue cancer, is driven by tobacco, and there is no incidence of HPV. HPV presence is there in the nasopharyngeal and all other different subsites, but not in the oral cavity. Even the ICGC that initially kind of like talked about, then ICGC kind of like changed its line and says that even if the traces of HPV DNA is present, it is not expressed. So we know that that these samples are HPV negative when it comes down to the oral cavity. I'm not talking about the nasopharyngeal aspect of HPV.
Thank you.
Sure.
Great. Thanks, Amit, for the wonderful talk and discussion. Maybe I request Lawrence to please hand over the.
You're killing me deep.
Further deep, yeah?
Sure, I will.
So, moving on, we kind of captured another, alumni of ICGEB who was passing through New Delhi around the time when we are holding this meeting. So, our next speaker is Abhinav Kaushik. He has worked with Dinesh, and he has done some excellent work, so he's going to share something with us. Yeah.
saying is that because in data science you don't have like protocols. There is no single protocol that like data. Anything can be right, anything can be wrong. And until you improvise, until you criticize yourself, there is no way out you can figure out the right way to write the data. So that's exactly what I learned, because I still remember I presented the data to my supervisor, and he said, go ahead. And then I started experimenting with my data, and eventually at the end, you know, it took some time, of course. It took many years to publish my first paper. But at the end, I learned a lot of things. I learned how to troubleshoot. So that's my lab. I mean, I had a lot of such selfies. And one more thing, like on the right hand side of all these weekly workshops we used to conduct every year. They are like tremendous. Like they were very helpful in gaining the experience and learning the things which on my own, not just for others, but for myself too. Okay, moving ahead. After completing here, like based on my profile, I published a lot of papers with the help of my supervisor. I in fact joined CAUS. CAUS is the university founded around 2009 in Saudi Arabia. I joined this university even before my Viva. And it was a great experience. Like this on the left hand side, this is the library of CAUS. It was one of the best, most universal library I ever saw. And then this is the entire campus. Like it's a different city, a different world on its own. Like it's a different experience altogether. And that was the team. And my supervisor on the right hand side, Professor Arham Pine. And it was a great group. I learned a lot, and those things I learned in the PhD, I applied here directly, indirectly. And unfortunately, I had to leave because of some visa issues. Then I started applying, and then I ended up here. This is a remarkable center. It's called Sean Parker Center for Allergy and Asthma Research. It's a unique center on its own, which has different domains. It's kind of a small institution on its own. It has research labs. It has the clinical trial domains. It has climate change, which is recently added. It has several hospitals within itself to take care of the patients. So this is headed by my supervisor, Professor Karina. She's the director of Sean Parker Center for Allergy and Asthma and the director of it. And this is my group. I must be there on the right side. So this is my group. This is like half of the team. So half of the team was not there. So it's a big group. So I take care of all the data. I take care of all the data analysis aspects of this institution. And I'm lucky to be here. Okay. Second good thing. I got to know about the Alumni meeting. The second good thing, and this paper came online. So I thought that let's present this one. So this came online this month. Second November. This paper talks about like CD8 T cells. So our group works on allergies, mostly peanut allergies or multi-food allergies. There are other domains also, but this particular project deals with peanut allergies. Peanut allergies are actually very, very severe problem in US. In India, we hardly heard about peanut allergies. There could be seafood allergies, there could be a lot of other allergies. But in US, it's a very big problem. It's all these nut allergies, like common allergies, cat allergies, different ways. And it can be deadly and it could be life-threatening depending upon how severe the reaction is. Okay, so this particular paper is based on a technology called mass cytometry. Some of you may be, or most of you may be aware of this technology, but some of you may not. presence of CD57. Now CD57 is a marker for terminal differentiation, which means that each cell becomes, they lose the tendency to multiply much, and they reproduce cytoplasm. They can interact. They would interact less, I would say, like in a very, very broad way. So what we observed is that high proportion of CD57 positive, CD57 marker within the effector memory T cell is a decentralized individual, as compared to the. Now coming back to the nice cell again, nice cell data again, we
Thanks, Aina. Excellent work. Any quick question? Okay.
Hello? Yeah. So I just had one quick question. Why did you stimulate, did you do a recall with PMAI rather than CD3, CD28 co-stimulation? That would have given you a receptor-based activation of the T cells rather than a pan, non-receptor based activation. That probably will activate multiple signals. So that's probably why your CD57 actually came, because it may not be that if you stimulate with CD3 and CD28, CD57 may not have come up.
Okay, thank you. So for you mean to say from the effector memory point of view? So the role of effector memory cells is still very controversial, I would say. And there are not many studies from peanut allergy point of view. CD8 cells is one of the least studied cell types when it comes to those. So we are the first one to propose some hypothesis when it comes to this particular clinical trial. And so we conducted multiple things and we just try to connect the dots.
Yeah, but what Rajan was asking is whether, so did you also use anti-CD3 to stimulate the T cells, or didn't do that? Anti-CD3 and CD8 to stimulate the cells or not? Only PMA was used.
So for other stimulated cells, we did use two stimulations only. One the peanut stimulated cells, in which we just stimulated the peanut, and the second with the PMAI.
Okay.
And for the PMI, we just it was a very high stimulation for sure, but not the CD3 and other stimulation.
Good. Any other question? Great. So let's give a big hand, and maybe I request Dr. Salunke to hand over this to Abhinav. Thank you. So moving on, we have two more talks from the alumni. And next speaker is Duong Ho, who is going to speak on microbial enzyme application in producing chitin oligosaccharide as a biocontrol agent. So again, I didn't have much overlap with him, so I don't know which lab he was. That's something which he'll going to probably tell him.
Because they are supporting. Okay, so good afternoon. So first of all, I would like to thank the organizers, general ICGEB editors, and due to the ICGEB director and all organizers give me the chance to be here. So I just came to ICGEB from 1996 to the early 1999. So at this moment I work in the PhD lab. So then I come back to Hanoi University. So because of this I work with microbiology. So I think that in my life and my careers, I spent found this time very fruitful for me, because of this time all the faculty and all the scientists from the PhD lab and from ICGEB is very kind and very helpful to my activity. So by this time I would like to take this chance to express my special
thank to the Professor Sukuri, group leader, and Dr. Sunil Mukherjee, my director, supervisor, and technical support from Dr. N.K. here, and Dr. Amos, and all of the members of the PMP Next. So today I come back here, so I just give very short talk about some of the topics I'm doing. So I try to find the combinations between the microorganism and plant. Okay, so the first So my talks concerning about some content, the prepare the course and about we check the activity of the photosynthetic function, and the other we try to check about from the in vitro and the field test, and the last one we check about if course can enhance about the plant immune system. So in this I'd like to show you that here Vietnam is a stream exporting, so anyone 10 billion is a big one. So I think that almost so Vietnam's among the 5 countries exporting stream, but in this about the byproduct, about a lot of the byproduct from the stream cell, but it's containing about the chitin, but from chitin we can prepare about chitosan. The difference is that because chitin contains both units glucosamine and acetylglucosamine. So when we acetyl group, it becomes chitosan. But the thing that chitosans in water solute, but chitin cannot, so very difficult to make it. So of course application we have many application, but here we are focusing about for biocontrol. So normally about people prepare the chitosans by the chemical way, not in Vietnam also. That's the many of the institute research group and even very popular. But many publications thinking of using biological method. So in this case, we try to use. So I suppose that we try to use two kinds of bacteria, Bacillus for protease and Lactobacillus for organic acid. So finally, I think that we can provide here, we can develop biological process. So in case of chitinase, we try to screen it. So two sources. One source from actinomycetes, and the other source from filamental fungi. So we try to check between the chitin from A from the actinomycete and B from the filamental fungi, and try to check its sigma 1. So by the native, you see about the profile different. So then, here in case of chitin deacidinase, we tried two sources. At first, we tried with some support, we tried to get recommended one, but it's not very strong because that's here is we tried to overexpress in yeast, PGF battery, but it's the gene come from rhizopath, but you know, so then we select one kind of streptomycin, so it's showing some activity at molecular weight of 45, but from rhizopath about 75 kilodalton. So when we try to prepare treat about chitin with enzymes, we get about two kinds of enzymes about chitin A and B. So here is about different about pattern. So in the case of the cos A, molecular weight it comes from the 300 to the from cos A from the 300 to the 1,200 Dalton. In case of the cos B, it's less than 700 Dalton. But now we come to check about anti-fungi. So body there is many several of the pathogenic fungi such as botrytis or fusarium or phytophthora. It's very harmful to agriculture. So we try to check here with about two levels. The first level about inhibition to spore germination, and second about the growth. So now we have two kinds of enzyme sources. We try to find out about the difference about the activity. So here's this slide and the picture show about the different about the concentration. So here we come to the results show that cos A inhibition is stronger than cos B on about spore germination of the botrytis cinera. So we try to check about two growth of the same fungi. So the same story, almost the same. Such a bit we try to about inhibition rate. We think that cos A is better than cos B. So they come to see that so far about chitosan or cos, there is a many report showing that about really cos have activities inhibit about growth of some bacteria or fungi. So depends on about mainly three about the molecular weight, concentration, and about dP. dP is that mean the degree of dd. That's the degree of the acidity. So in our case, we try to find that if the pattern of the cos different between two kinds of cos A and cos B when we use a different source of the enzyme. So here in this slide we can see that about the different about the unit of the glucosamine. In case of the cos A, it's much more than about the cos 2. But about the pattern, we check from the degree of polymerase from the 2 to 10. Because normally many reports set up about the degree of polymerase from the 4 to 14 shows activity better than about the bigger unit. So now you come to with about protecting plant from the fungi. Here we experiment, we try in treat with without treat with the goat and with treat with goats. And here in this case we using chitosan, conversion chitosan with degree of polymerase around 300, quite a big one. So in this case here, we can see that about in case the plant here we use tomato. So it's the goats. The goat we just using the goat egg, but the goats protect the potato from the infected by the botrytis cinera. Very clear. So here's about the rate of protection rate. But when we try to treat dye with about Cancun can see that in case without treat with cold, it clears that plant damage. But in case of treat with cold with damage very less in comparing, you can see. So now on the fintech, we try the fintech about our research. So here we do not have a greenhouse, but we send to the other institute. They work with the fintech. Here we try with many kind of in Vietnam fusarium or with some of the Phytophthora or botrytis, or so you can see that in comparing with some of the commercial fungicide, you can see that the ratio is quite a very good one. So we come to the conclusion that cost preparation can showing strong efficient the fungicide, in comparing to the commercial product. Now, so the question is that cost showing that have some inhibition to microbe, such as the fungi. But now the mechanism for in case of the cost prevent fungi germination or growth not yet clear. Have some reports said that maybe cost is about positive charge. But in case of might be involved about the replication process, which about negative charge, might be, but that's still not clear. But in case of plant, if the coach can enhance about plant immune as an anicide, so we try to do some experiment on this. So here's because now there is some reviews mentioned that there is a two systems of the plant resistant to the disease. So one about the system is acquired resistant, relating to the phenols compound, and some the other resistant protein such as chitinase, some enzyme chitinase or glucanase. So in case of examples here, we see there are some of the key enzymes such as PAM, that means phenylalanine, nanins, ammonious nitrates, and PPO, that's polyphenols oxidase. But in the other system about IFR, that's induced systemic resistance. So it's related to the several kind of lipids. It involved about the synthesis of some hormone for the resistance. is called in short about NIP. So the key enzyme is lipoxygenase. So now we try to check about this enzyme or some expression of some resistant protein. So here, so in case checking about some enzyme, so you can see that in case of control here with about the cytosine, here indicator of about the cost. So in different whole plant or some the leaf, so different you can see that in conclusion that okay, so about cost enhancing spam and PPO activity that belong to about systematic acquired resistance, but not enhancing about the loss enzyme. So in about induced system mixture resistance. So one more experiment we tried here to check about expression some enzymes involved by using about quantitative real-time PCR. So there is 3 gene we use about protein resistant 1, 2 that have some glucanase activity, and protein resistant 8 relating to about chitinase. So we can see that cost enhancing about expression of those 3 those resistant protein. So come to about this slide, still there is some report and hypothesis reports by NUS and 2005. So chitin or the cytosine have some of cost, so we come to through this way, activated with some receptor. But more than that here, you can see that through the resistance mechanism, that's about the hypothesis. So in case of SARS on ISA, so in course, because we infect about the whole the stem and the leaf. So in this case, enzyme here, PAM, and about the resistant protein about the enhancing. So that only the effect have some enhancing about the mechanism SARS, but no with the in case of induced system is resistant way. So that's about we come to the end with the conclusion that we developing the biological methods for producing cost, and we can check about because the difference that the code activity not only depends on about concentration or degree of polymerization and degree of the acetins, but also depends on about pattern relating to about chitinase source using. And second, that we check about both in vitro and in vivo and FinTech confirm that it can be used very good about biocontrol agent. So of course here cost really is unedicited enhancing about the plant's immune system through about as your way. Thank you. Yeah.
Thank you, John. I think we are now really pushing into the end of the today's sessions. So thanks very much for taking us through what you have been doing. So I would request Dr. Salunke to hand over the memento. Thanks. So our next speaker, also the last among the alumni who is going to speak today, is Tatiana Maria Lopera Mesa. She was immediate senior to me when I came to ICGEB in 2002, and so we had really fond memories of our time during PhD. I understand there is a lot of emotions and excitement for this session to get over and meet outside the hall, and I can see a lot of people are still outside the auditorium, but I would urge them to control the emotion for some time and hear out the remaining sessions which is planned for the day. So without taking much time, let's give the session to Tatiana, and she will take us through a very, very interesting title of Life Science and Chai, which she would experience in India when she was here during PhD.
Okay. Good evening. I know you are all very tired, and I hope I will not take much time to go through this presentation. First of all, I want to thank for this opportunity that you created for the alumni to come together. It's really a great moment for me to be here. I never imagined that I will be visiting ICGEB again after 20 years, so I'm very excited about this. Okay, so I will not have much results about my research. This is going to be more I'm going to tell, it's like I'm coming home and I'm going to tell you what I have been doing since I left, because I feel that you are like my family, so that's what I'm going to do. But first, I will give you a little bit of context about who I am and where I come from. Okay, it's not working. Okay, yeah, so this is a brief outline of my presentation. I will go through this again. So first, a little bit of context about myself. I grew up in a city called Medellin in Colombia. Normally in this side of the world, people are not that familiar with the geography in South America, so I just show you again where is Colombia and where is my city in Colombia. I grew up there. I did my graduate student studies, master degree. At the time I was graduating, there was not much postgraduate education, and travel abroad to get postgraduate education was very rare. So, I mean, I am one of the people who had this good opportunity. My first contact with ICGEB was through a workshop. So, it was in 2000. And then in 2001, I joined the PhD program. I was the only Latin American student, so it was I felt quite different from everybody else here. Sometimes I felt that I was in another planet, doing PhD. So my thesis work was at the malaria group. I was supervised by Professor V.S. Chauhan, and my mentors, direct mentors in the lab were Dr. Ashima Kuswaha and Dr. Asif Mohammed, who is here. I work with this nice protein called MSP9 from Plasmodium berghei. And more than whatever I got with this protein, this means a lot for me, because it was the protein that allows me to learn what I learned on the PhD, during my PhD. So I'm not going to explain much, but this is a protein that is important for the parasite, for the invasion of the parasite, and at that time it was one of the possible vaccine candidates to include in multi-component vaccine. So yeah, while we were doing PhD, we also had this tradition of going for chai in the evening, and many of the people of that time, we got together at the canteen and we discussed science and other things. So I remember those times, and I was missing having chai in the evening for all these years. So, well, the time was passing while I was trying to get the recombinant protein to get refolded and purified without getting precipitated. And while that happened, I built good relationships with people and good friends that are still here. Some of them are here. Some of them could not make it, but still we remember all of them. I had some nice time in the lab, and also I did hard work. We managed to have some few parties. I don't know if you recognize this guy. He likes to dance when the proteins get purified, and I also got to interact with my batchmates and with my junior students. So anyway, that was life at the PhD, during the PhD. And eventually I got some protein to keep working. So we found that, we could show some activity and functional activity of this recombinant protein. And as I said, more than the results, it was my process. So for me, this protein was important because it allows me to learn what I learned at ICGEB. Finally, the challenge of writing the thesis. And here I also need to thank again all the faculty, Professor Chauhan, Asif, PM, many people in the lab that helped me, because I was this foreigner who needs to write a thesis in English. My English was not that good. The senior students, all the group of friends, my classmates, friends in general, Chanaki and Janet, who were very patient with me. And this is the time to thank them again. Eventually I managed to write the thesis and I got the degree. And then I move on. So we published the paper. And more important than the result was the process. So I realized that when I reflect on what it means for me, I realized that was the time when I learned planning and critical thinking. I also developed technical skills, of course, but I learned from the daily basis experience how to troubleshoot in the lab, how to discuss work and ideas, and that's what you learn only by spending time in the lab with the colleagues. That's what I am grateful for, the experience I have here at ICGEB. So then I moved to a postdoctoral experience where I had the chance to work at the Laboratory of Malaria and Vector Research at NIH in the United States. And I was glad, I was lucky to be involved in this project where we study innate resistance and acquired immunity in children exposed to Plasmodium falciparum in Mali, West Africa. The principal investigator of this project was Dr. Rick Parhurst at this lab. And well, as many of you may know, falciparum malaria is a big problem in Africa. The main vector is Anopheles gambiae, and specifically in Mali, the malaria season goes from June to October. And there, around 70% of children carry at least one red blood cell variant, including hemoglobin S, hemoglobin C, alpha thalassemia, G6PD deficiency. And each of these variants are known to confer some degree of protection, but studies are not always conclusive, and I mean, it's expensive to carry out powerful studies to understand how all these variants combine to get protection to children from malaria. This is just to show how in this population some children have more than one of these polymorphisms. So it's interesting to think how the combination of these polymorphisms may work to confer protection. So that's how I was involved in this prospective cohort study that it was run from 2008 to 2011, where we had the chance to enroll around 1,500 children of 6 months to 17 years old. And the main objective of this study was to determine the relative risk of Plasmodium falciparum malaria in Malian children carrying different RDC polymorphisms, and keeping in mind covariates that we know may influence this outcome, like age, sex, ethnicity, or the village from where they come. So just to go through the outcome of this study, so we managed to enroll 1,500 children, and in this population we record around 4,000 episodes, malaria episodes, diagnosed over 2,656 child years of follow-up, because every year we were working on a specific village in rural Mali. So practically the whole population of the village was enrolled in the study. So the same kid could come every year with a malaria episode. So we had to design a unit to measure this outcome. And there we found different proportions of frequencies of these RBC polymorphisms. And I mean, the main outcome of this was that malaria incidence was reduced, again, as it has shown before, by the heterozygous state of hemoglobin S compared to normal hemoglobin. And also G6PD deficiency was a protective factor in girls, homozygote girls, compared with heterozygote, with normal G6PD girls. We didn't find any significant change in malaria risk by alpha thalassemia or ABO blood group. And we also, although there was a tendency, this is just a cut of the whole table that you can find in the paper, but although we have a tendency of interaction between alpha thalassemia and hemoglobin S in terms of the risk of malaria, this interaction was not significant to change the risk of malaria. The interesting thing, or the most interesting thing for me, is that we could, with this study, we could show that the protective effect of hemoglobin S was, sorry, what did I do? Okay, oh yeah. So the protective effect of hemoglobin S was specific of age. So in this population, we found that hemoglobin S was a protective factor in children between 1 and 7 years old, with the main point at 3 years of age. But after that, children older than 7 years old, that protective effect of hemoglobin S was not significant. And this has to do with the effect, during this time, children are building immunity, acquiring immunity. So we think that in real life, what happens is that at the beginning of the life, hemoglobin S protects children, but then immunity takes over. And then when you try to measure the protective effect of hemoglobin S, you cannot find significant difference. And we also found that the protective effect of hemoglobin S has to do with reducing parasitemia. Although this reduction is not that high, it can give a clue of what could be the mechanism of hemoglobin S to protect children. From that study, so the main conclusions were that hemoglobin S reduced both the risk of falciparum malaria and the parasite density at the time of clinical presentation. The greatest degree of protection conferred was in early years of life, and homozygous G6P deficiency encoded by X-linked A- allele reduced malaria in girls. In this cohort, we also had the chance to carry out a parasite clearance study related with the response to artesunate, and also evaluate the effect of age as a surrogate of acquiring immunity. So we set up this clearance study in a sub-cohort of 200 children, where we were able to measure parasite density. These were the inclusion criteria for the kids. They were treated with artesunate and amodiaquine, and we measured parasite density pre-treatment and post-treatment every 6 hours for 48, and after it was undetectable for 48 hours. So in this study, we use this measurement that is called half-life of parasite clearance. That it has the advantage of taking, measuring the change in parasite clearance, the time that it takes for the parasitemia to drop by half in this portion of the curve. And the advantage of this measurement is that it doesn't get affected by the initial parasite density, because when you do these kind of studies, you need to take care of all the possible variables that affect your outcome. So in this way, we were able to have more objective measurement of how the parasitemia changed over time after treatment. And again, we did a multivariate analysis where we found that from all the host factors that we could measure in this population, only age has a significant effect on the outcome of parasite clearance. So again, in this population that is exposed to so many episodes of malaria, it's important to keep in mind that age as a surrogate of acquired immunity may interfere with the outcome, and you need to keep that in mind. So we saw that there was an inverse correlation of the half-life of parasite clearance with age. We could measure that 4.1 minutes shortening of half-life for every year of increasing age in this population. And shorter half-life means faster parasite clearance. So it shows how the age as a surrogate of immunity may be altered these measurements. So yeah, in this study the conclusion was that acquired immunity could be related with reductions in half-life of parasite clearance in children with uncomplicated malaria. And yeah, so you need to keep that in mind when you do these type of studies in hyperendemic areas like Mali. So this was done by a very big team of colleagues in Mali, and also the team of work in NIH. I just go through this because I know you are very tired now. Okay, so on the backside of these signs and results, I also had this experience in Mali where while I was working and doing things in the lab and culturing parasites, I get to know this person who taught me the instrument, this instrument. And the interesting thing was that he didn't speak English, I didn't speak Bambara. We managed to communicate somehow, and he managed to teach me the instrument and managed to learn something. We even record a CD. But overall, the experience was, and I think something of what I experienced here in India helped me to have this also, because when I was here, I realized that I had to be very open to people from different cultures, from different languages, different way of thinking, and the time I spent here helped me to do that. So when I went to Africa, I realized that, okay, I'm open to learn from others. I can explore how they think, what is important for them. And for me, this relationship in particular was very meaningful. He died after some time, but I want to remember that because it's part of my life experience in Mali. And of course, I said that chai is part of my life. So in Mali, there was also tea, like an African version of chai. And I also used to have tea there. Okay, so finally in 2013, I went back to Colombia, and I joined as a faculty at the School of Medicine at Universidad de Antioquia, and I am a researcher at Malaria Group. Just a brief introduction to the university. It's one of the oldest public universities in Colombia, and so this is just to take you through different studies that I have been guiding. So I am not going to explain results, but I just want to share with you the type of study that we have been doing related with platelets and Plasmodium vivax, artemisinin resistance, and also diagnostic tools, improving diagnostic tools for malaria, or in collaboration with this was in a collaboration with a company in Japan, ICGEB. Okay, and most recently I have been interested in humanities because I realized that my role as a faculty at the School of Medicine, it needs to get more connected with why are we educating students to become medical doctors, what is really important. So I came up with this colleague, John Borges, who is a medical doctor, And we thought that we need to design a course that help us, that help the students and help us to be able to talk about what is there to learn or to become a medical doctor. So we create this elective course called Humanization in Medical Practice. We draw the course in a napkin. Once we were having one beer and we decided, okay, we are going to have a course with these components, and we signed that napkin. We still have the original napkin. And then 1 year later, I wrote up the program. We got it accepted at the university as an elective course. And then, we have been offering the course since 2017. It's always fully booked. The students enjoy just having that space where they can talk and they can express things about their experience as a medical students, and also how to prepare for the practice in professional life. We feel that we have the chance to have a real human link generated among participants, students, and teachers, although we don't feel that we are really teachers. We are just facilitators of this place. And it has been a nice achievement. And finally, I recently got also interested in scientific mentoring at postgraduate level. So I started asking myself this kind of question: what does a mentor teach to a PhD student? In reality, what do you teach? And what does a PhD student learn from his or her mentor? How both of them experience the PhD process and the relationship, and how this experience impacted the doctoral education in biomedical sciences. Because I feel that we all do a lot of science, but I don't know to what extent we really ask ourselves the human side of this. And I think that is also important. So I set up this project, the pedagogic relationship between mentors and mentees, at a basic biomedical science doctoral program at the University of Antioquia. And I have been moving towards qualitative research. So I have been doing interviews to mentors and mentees about their experience at a PhD level. And I'm getting, the outcome will be discourse analysis through this paradigm of symbolic interactionism. So I hope to be able to comprehend better what happens there that is so critical for the education of new scientists. Finally, I just want to express what ICGEB has impacted on my life and career, and I feel that is something like open my view of the world through a small window where I can look now with different eyes, and the fact that I could come here and tell you all this is really meaningful for me. I feel that this is like India is an old friend who called me again for a visit after 16 years, and I have to thank ICGEB for creating the opportunity. Thank you.
Thanks, Tatiana. I think we all agree that you had excellent, we have had a very wonderful journey so far, and wish you all the best. You also re-established one of the old ICGEB ideas, that a lot of interesting ideas evolve over beer. So that is something which we all really agree with. So is there, yeah, Sanjay.
So, hi Tatiana, I'm Sanjay. Oh. So somehow we miss each other many times. So I was at ICGEB. I was at ICGEB from June 30th to 15th October 2000. So you came after that. Then I was at NIH, I was at MBDU. I'm one of the part of establishing the Mali site from 2005 to 2001 to 2005. You must have stayed into Bob Say, the guest house.
Yeah.
So, and I left NIH in 2006. So Rick Ferres, Carol Long, Kazutayo, all were the colleagues on the other side of the road.
Okay.
Only thing now we are building a big plant in Medellin, beautiful city. I hope I should see you there.
Yeah, okay, wow. See,
So this,
everything is connected.
somehow. So I'm thankful to organizers. They called me and we can remember because Mali site and Burkina Faso both we built. And sometime I was caught by the terrorists. So I used to show my Indian passport.
Okay.
So,
Yes.
but you have a really vast experience working into the research lab and then going to Mali and working in the field. That is, I have experienced by myself, it's life changing. So nice to hear from you. Thank you.
Thank you.
Thanks, Sanjay. So I'll request Dr. Salamke to present this to Tatiana.
What a great way to end this session. Fantastic.
So now we'll move on from alumni to people who are involved at ICGEB to establish a much bigger platform to have alumni connected or staying connected with ICGEB, in a more formal setup. So I'll invite Suzanne. She is at ICGEB headquarters in Trieste. She has been doing something with respect to, setting up the overall platform for ICGEB alumni, involving all three, components of ICGEB. So she had something to share with all of us, and let's hear her out.
Hello? Great. Thank you very much for that introduction. So I'm based at ICGEB Trieste, Italy, and while I'm not an alumni, I feel as though I almost am. I've been associated with the ICGEB for 30 years, and it's a great honor for me and a real delight to be here today with my colleagues from New Delhi and with all of you to reignite the Alumni Association. Truly momentous, I think. So, sorry. So, as a science communicator and as someone who's trying to build bridges, I've asked, well, what has ICGEB done to promote science development? And when I look out into the audience, I think that that's very apparent, and from what we've heard today, it has contributed to quality education. And we've gathered some numbers. We've recontacted 3,500 alumni from ICGEB. That's PhDs, postdocs, long and short term. We've been able to build up a database of 1/3 of these who received the newsletter of this meeting yesterday, in fact. And they're in over 60 countries, and we estimate around 750 PhD graduates. So how have we tried to collate this information? We established a web page on our website calling our alumni across the globe. And on this page we feature testimonials. We also have an interactive map which shows where the alumni are. You point your cursor across the globe and you can see which institutes they're at and how many alumni we have there. And as an alumni, you can access the reserved area on that page and also receive a list of the contacts and the addresses of those people. So we're really trying to build up connections across regions. So from a cohort of just over 400 students that we've reconnected with and who've reached out to us also, we can see where our ICGEB alumni are today on the job market, in their positions, and around the world. We're still trying to work out how many have gone home, how many are elsewhere, but we're getting there, so we're building up that information. So we also reach out through newsletters. We've just launched an alumni newsletter, and everybody can sign up on our website. We also run seminars and podcasts, and the current students really like to hear from our alumni on science, but also on mentoring and on soft skills as well, as they search for their path in life. We also run engagement activities. This is a competition that we hope to launch across the 3 components, open to all the current students, for a science communication internship. This is hosted at the University of Cambridge by Dr. Chris Smith, who's a clinical virologist, a journalist and a science communicator, and the founder of the Naked Scientists. It's a BBC 5 program. He effectively reaches about 500,000 people a year. So it's a great training and a great opportunity, and he's connected with the ICGEB now. So as part of our international seminar program, I'm sure we do many of the things that many of your institutes around the globe do. We're now counting the number of alumni seminars we've given. There are approximately 50 seminars held in the 3 components throughout the year, and these are live and in streaming, and then collected on our YouTube channels, on Apple Podcasts, and on the website. And collectively, we reach almost 100,000 people a year. And it's interesting to see that the majority of watch time is actually in countries that may not necessarily be ICGEB member states, but that means that your science is really getting to people. We also obviously follow up trying to imbue a culture of storytelling. And I'm very grateful to many of the people in this room also for lending their time and their stories to the videos that we have on our channels. Many of these are done in-house, and more recently we've also done a project for a donor, the United Nations Office of South-South Cooperation. They funded 5 fellowships. One is for Rejwana Najneen, who is here from Bangladesh, and 4 others who are in Cape Town. They've asked us to prepare professional videos. The exciting thing is that they're going to launch these on the International Women's Day, 8th of March next year, at the UN in New York. So I think that gives us huge visibility and hopefully opens the activities of fellowships up to other donors also. So on social media, we're hugely active, as many of you are. My colleagues both here and in Trieste and in Cape Town spend a lot of time, and they're very patient with all of you, and likewise you're very patient with us, sending us photos, keeping us posted, and then we can share these on the alumni groups that we've created, both on Facebook, which has reached almost 500 people in just 1 year alone, and on LinkedIn we have a public of about 21,000 people, so it's really it's important. So these are some of the numbers on our analytics that the activities of the alumni are incorporated into. So 1.2 million visits to our website each year, 30,000 readership of our newsletter. Our outreach on social media, I think, is quite phenomenal if you consider that 2.4 million people we reach through the collective efforts of social media. So it's certainly an avenue that we continue to pursue. We also are grateful to our alumni for their testimonials in our publications and for taking part in our media relations. And so basically we're all doing this to create an international network of enterprise specialists, and that is you all. So please visit our webpage, the LinkedIn site, Facebook, write to us. Thank you, and thank you for this opportunity to be part of this. Thank you.
Thanks, Suzanne. Really wonderful. In a short time, you captured everything that is being done to really formalize this whole ICGEB family, I would say, extended over different part of the world. So thank you very much. as we now move towards, I would not say final, because final would be the the meet and greet and dinner. So, so the final formal part here, which is again, I would not call it formal because we'll keep it informal. Some ideas, thoughts, free-flowing ideas around this whole alumni meet and a future course, not only for alumni but overall ICGEB as an organization that people have been thinking around. So we'll move on to the panel discussion, and as some arrangements will be done, which I'm sure PM is taking care of. What also is something which didn't occur to us because we were doing too many things while organizing this meeting. It was impossible to have all the wonderful alumni who have come to ICGEB today to have them all speak and give a talk. That was something which probably would require a 3-day or 4-day session. So we had some of you speak here, but there are a lot of people here who have gathered. Some of them know each other very well. Some of them probably haven't overlapped here. So it may be a good idea to quickly have even more lightning talk than the kind of talk the students gave. And rather than coming here and setting up and taking a little more time, I have the list of people who have come here. So if we can just stand up and whatever comes to our mind, we just speak. So, right, whether it is where you are right now, thanks. Whether you are right now, or what you feel about ICGEB, or what you think about what ICGEB should be in next 10, 20 years time. So I would need some help with someone who can do some running around with the mic. So one is with Mayank that side, and someone can take it. I will speak from here. Yeah. So the list is alphabetical. So I'll begin with Anand Srivastava. He is there.
Anand, please stand up.
Oh yes.
From the top.
The same way as we did yesterday. Right. After that, we can have the group photograph. Yeah. Yes, Anand.
Yeah, I'm thankful to all of you to organize this wonderful meeting. I'm Anand Srivastava. I did my PhD from here, as you so I was working with Dr. Chetan Chitnis from 2002 to 2008, and Dheeraj is my batchmate. And after that, I went for my postdoc at Institute Pasteur. And from there, I went to the newly established Institute of DBT, National Institute of Animal Biotechnology. And I'm working on animal related diseases. Right now I'm working on a disease called theileriosis caused by parasite Theileria. And also I work on vector which transmit disease, a tick. And yeah, I'm very, very, feeling very good to come back to ICGEB after a long time. Thank you very much.
Thanks, Anand. Next we move to Puran. Hi, is there only.
Hi, I'm Puran Singh Sijwali, and first of all, thanks a lot to PM Sir, Dr. Salunke, Dheeraj, and all others who put together this wonderful meeting and gave me opportunity to meet with my colleagues and juniors, seniors, all that. So I finished my PhD from here in 1999 with Dr. Chauhan and Dr. Praveen Malhotra, and it's great to say that I happen to be the first PhD student from here, and there is one more Vishnu who is not here. So currently I am a faculty at Center for Cellular and Molecular Biology, Hyderabad, and I'm still stuck with malaria parasite like Dr. PM Sir, and I work on ubiquitin proteasome system and autophagy, and will be glad to talk to you more about it whenever we have chance. Thanks a lot.
It's a great way to say that you are still stuck with malaria parasite. So, Okay, next is Noah. I know I met him. Yes. he's there.
Thank you. Good evening, everyone. I'm Noah Machuki. I am a scientist at the Kenya Medical Research Institute in Nairobi. And I think I'm the youngest alumni here. I'm lucky that PM has the oldest alumni and I'm the youngest. Yeah. And I must say that we have crashed with Puran a number of times in our studies, and I think we need to collaborate and going forward. Thank you so much for the opportunity, and I'm looking forward for more collaborations and interactions. Thank you.
Thanks, Noah. Next is Dr. Gayatri.
So good evening, everybody. Grand to be back. I'm probably a hybrid because I moved from ICGEB when Dr. Sapori was there, and then I spent the rest of my time here. Very fruitful, learnt a lot from everybody. It was a great environment. I currently work, and I've been working since I left ICGEB at the MS Swaminathan Research Foundation. So most people here have a perspective which is hugely scientific, very academic or industry. I work at an NGO. They work in sustainability science. So my work straddles somewhere in between. I'm specialized in abiotic research in extremophiles and wild rice species.
Thanks. Next, of course, we heard from Amit Dutt a lot about, but I think we should hear from Punjab himself. Mike.
Hi, good evening. I am Punjab Singh Malik, and Amit has made my situation really difficult actually. So I followed him. He was my senior in college, Venkateshwara, and did my summer project in South Campus Biochemistry Department. He was my senior there too. I kept following him, came to ICGEB in 2000, finished my PhD in 2004. So for students, let me just tell you, so the big difference we felt at ICGEB at that time was, especially if you come from a university setup, the open labs, and this culture was amazing. The environment we had, you can go to the any PI, sharing of resources, not only instruments and chemicals, everything. So I think I was kind of a sports quota person entry in this PhD program. So in the interview committee, Dr. Anil Tyagi, professor, and later I think he was vice chancellor of IP University. So he was one of the members. So as soon as I entered, and he said like, oh, he plays cricket and all that. And I come from a small village, and I thought this is the worst he can do. So, but maybe Professor Chauhan, he was also into sports, or something happened in that interview, and they selected me. So I had great time, and not only me and all our batchmates, our seniors, Yash is here. So we all had wonderful time at ICGEB. And for me especially, because I was with Dr. Mukherjee, so all my batchmates, and they will come with their scientific problems. And maybe I didn't know the project, but I had this feeling at least he knows what we are doing. So I had this, raised collars all the time because I was Dr. Mukherjee's student. And amazing person he is. And even now I went to meet him, I think a month back. And every time I go to his place, Madam Mukherjee, she is so nice. She will, have all different dishes on the table. And I'll bring one of my friends, like Jeetu or somebody from South Campus, who can, Das Radhi, who can actually talk to science. Talk science to Dr. Mukherjee so that I can eat whatever it is. So Dr. Mukherjee is such a passionate person about, he, for 2 days he goes for dialysis, and rest 3 days he goes to IRI. Amazing person he is. So I'm blessed that I got this opportunity to be here at ICGEB. And all these friends, Tatiana, she is here. So this Latin American white girl, and she will bring, salad bowl, what else, watermelon and everything. So me and Das, we will eat her breakfast, and then we will order this samosa and all that. I'm really thankful. I mean, the director, Dr. Dinkar, Pawan sir, everybody who is associated with this event. You made us felt, very special by inviting us, and I'm sure in coming years we will do many more meaningful meetings. So thank you so much.
Karthik.
Good evening, everybody. Myself, Karthik Pradhan. I am a staff scientist at NIH, USA. I did my PhD here with Dr. Shahid Jamil between 2003 to 2008. And I'm sad to see that he's not here. So many of the old faculties already, they are gone, retired. But I can see the reflections of my supervisor through other old senior faculties. And I'm very proud to be here. So let me tell a little bit of my experience here. So I came from Sambalpur, Odisha. It's a very remote part, and I was the first student to come from Sambalpur. And when I first time walk in, the first impression of the landscape of ICGEB really inspired me, and that was like a surreal experience. I thought I can never make it to, but in the interview they asked me, there were like Raj Bhatnagar sir, Amit sir, Chauhan sir, and they asked me, where is your thesis? I said, I come from Sambalpur and I don't have any experience on research. And they asked me many questions, theoretical, experimental, and somehow they believed in me and they selected me. And the first day I remember I was taught under Dr. Shahid Jamil how to pipette like 16 microliter, 213 microliter, this kind of things I learned. And I can tell you that when people talk about alma mater means the nourishing mother. This is my alma mater. Thank you, ICGEB.
Richard.
Great gratitude and humbleness that I would like to extend to people who thought that they should invite us. We belong to the same batch of 2000, known as the most vocal and the boisterous batch that I think ICGEB ever had, in terms of what we used to, the amount of knives that we used to create in the canteen and in the hostel. And there are stories, I think, which are built around our batch, which would be hard to replicate by any batch that has ever been produced from ICGEB. So in 2000, I think in March, we did a modern take on Mahabharata at this very stage. Okay, and that stuff is of legends. Not a single PI was spared. And we were reckless. This was first year of not even 6 months into PhD. People were mimicked with their dialogues and commented upon, but the graciousness of the audience was they got a standing ovation. Okay, people were there from all walks of life, and I was a Delhi person. I have been in Delhi, but ICGEB was, I had walked into ICGEB when I was doing my master's at JNU, and I had come to meet Professor Sapuri. He was then on sabbatical from JNU. And the first day I walked into plant molecular biology lab, I said, agar India se kahin PhD karni hai, to is jagah se karni hai. That was the view of the lab, There were glass on both the sides. You could come out of your room and see what we were doing. And most of the time it was not good stuff at all. Okay, absolutely not. There were matches which had to be hung. So let's go back to our director. So we told him, World Cup hone wala hai, TV nahi hai. There was no cable or this thing, He said, what is needed? हम लोग को cable channel चाहिए. He said, what do you want it for? All sports channel. Obviously later on became anything else. We didn't have canteen, so we got the canteen upgraded. We fought for washing machine. Washing machines were bought for us, right? We bought for washing machine. Then we fought for badminton court, okay? So there were things that we used to do. Then one day we went to Tina and said कि ये library कैसी है ना, सिर्फ science है ना, यहाँ कुछ भी नहीं है. Then I got a call that Dr. Chauhan is asking for you. And then I went in and he said, oh, you're that tall one, na? Okay? What do you want? I said, we need some books, some 100-odd books, which have nothing to do with science. So he said, okay, I'll give you 15 days. You come up with a list. So on one angle, it was badminton court. We had gym. We had table tennis working on the first floor, where you could hear the ping pong balls going every time of the day. And then we made a list of 100 books. And they were bought. They're still in the library, I hope. Okay, the best of books were bought. So ICGEB, I think, has been life-changing. We made friends for life, and hopefully we are still learning from them. We don't meet so often, but every time we pick up a call, you would get a response that makes your day, if not your week. Thank you so much for having me. So after finishing my PhD in 2004, I did a brief stint at at ICGEB. Then I went to University of Hyderabad. Then I was at Cornell for 3 years. 13 years I served at, and we established the College of PG Studies at Northeast, at one of the PG colleges on the lines of IRI in Meghalaya, where we have been mentoring students from Northeast and have been sending into agriculture system ever since 2009. And since August 1st, I have joined at ICRISAT as cluster leader for cell molecular biology and trait engineering. Yeah, thank you for having me.
Thanks, Richa. We all got benefited out of the fight you guys did. So next, Siddharth. I saw him, but I don't see him now. So next is Abhishek. He was also there. Ananda.
Hi, thank you for inviting me. So I joined ICGEB in 2005 from Dhaka, Bangladesh. I have completed my PhD with Dr. Sneh and Professor Sopodi. And since 2012, I'm at South Asian University, New Delhi. Currently, I'm working as Associate Professor in South Asian University. So in my training in ICGEB, whatever I'm today actually, all my research training is in ICGEB. I was a very raw, naive student when I joined here. And I was the first PhD student of Dr. Sneh. So I have done my postdoc in University of Western Ontario, and I have a brief stay at Germany, Jülich. But this is where my research training, and this is what made me what I am today. And I have seen many students of South Asian University today who have done masters with us and currently doing PhD here. And I'm really thankful to ICGEB, and whatever I am today or whatever research I'm doing today, my training has been here. Thank you so much.
Thanks, Ananda. And because of his really hectic schedule, I need to invite Shishendu Mukherjee to quickly have a word with all of us.
Thank you, Dheeraj. When I first, Dinkar sir told me that we are going to have an alumni meet, and his invite came to me along with the PM and Dheeraj. I was literally feeling very nostalgic, because I joined ICGEB somewhere around 99-2000 and was here till almost 2008-2009. So get an alumni invitation, I was literally privileged, and it was feeling nostalgic for me, because I came to ICGEB from an industry, and I was handling the IP and technology management. Later on, I started working on the PhD program, and many of our PhD students have been one way or other connected and still connected to me. ICGEB was a place which played a very critical role in our career development. And it has made us actually, I am literally proud to say what we are today, at all we have achieved in the life is because of this organization. Why? Because it has taught us how to address challenges. You know, all the PIs which are sitting here at that time, I think a lot of them are here, PM is here, Dinesh, Shiva Reddy sir, Naveen sir is sitting there. I think MK, Suresh I saw sitting somewhere. I think literally they handholded us, and we learned every day new things happening out from that. And that has made our career. Especially working with the young PhD student, I still remember Dheeraj, when he was coming for his first interview, he walked in and he asked me what is my roll number, why I think he sat in this hall to write his exam. And then he was, and not only Dheeraj, I think a lot of you, we interacted, and I was the first one to give a lecture to them what is about ICGEB. And then I used to take them to each lab and show them this is malaria, this is PMB, this is recombinant gene lab. That time that was the nomenclature. It literally, and then the classes used to go on, and then the allotment of guides, that was the beautiful thing. Somebody came to us, Ryan, I want to change. Somebody was so I think this literally helped us, help all of us to grow. Nothing to say. And one more important thing, Dinesh is sitting here, he was my canteen buddy. So Dinesh PM, we used to be in the canteen committee, and I think the best canteen in India some institute has is ICGEB. I still miss the canteen and things like that. Apart from the canteen, the intellectual discussion we used to have during the canteen session, the tea sessions was really great. But I think finally ICGEB made us what we are, and literally respect ICGEB, and we will never forget the mentorship provided by ICGEB, by the the PIs here. The students' interaction was really so enriching that we used to learn a lot of things. As Richa just said, ये चाहिए, फोटोकॉपियर मशीन का भी एक चैलेंज होता था तब, रिचा आप भूल गए, फोटोकॉपियर लाइब्रेरी में नहीं होता था तब. हाँ, अच्छा, फोटोकॉपियर आया सर. So I think that all literally made us but it is very interesting, and thanks Dinkar sir, PM and Dheeraj for the invite and bringing us here today all together. Thank you very much.
Thank you. I see Siddharth is back, so.
I just want to thank everyone for the kind invitation, especially the director and Dr. PM and Dr. Dheeraj. Right? I spent the best part of my life here, made the best quality of the friends, did the best amount of science. Whatever I have done, it's because of ICGEB, and the worst part is I go out and I look for ICGEB everywhere. And that, unfortunately, I will not be able to find anywhere, because it's only in ICGEB you get what ICGEB is. My boss, when I joined him, was a month old. He didn't have any projects, but my work was always on time. Thanks to PM sir, who doesn't know that I post most of the kits out of his lab. And Dr. Shahid sir and Dr. Vijay sir, namaskar sir. And we had the best time because everybody was so cool and still is. And whenever we ask for help, they're always kind enough to give. Thank you.
Thanks, Siddharth. Shweta Sharma? Left? Okay. Rahila Sardar. Okay, she's there.
Hello everyone, I am Rahila and I recently did my PhD with Dr. Dinesh Gupta, and after my PhD my next task was either to go for a postdoc or what next. Then Dr. Dinesh suggested me, Rahila, instead of going for postdoc, I suggest you to start something of your own. Then I started my own venture. Recently, it's just 7 months back, that is V Genomics, where I'm providing genomic services, different kind of genomic services, especially focusing on the preventive healthcare. And the next challenge was to have a team. Then Dr. Dinesh suggested me, yes, there will be lot of challenges for you, but if you try, you can do. And now in my team, I have an expert with more than 35 years of experience from United States. My co-founder is from US with 150 patents. And we have a team of clinicians from different organizations like AIMS and Fortis hospitals. And just in the 6 months, we are getting so many collaborative projects from prestigious institutes like AIMS, Fortis, Eye hospitals. And I'm very thankful to Dr. Dinesh that he suggested me to start this kind of company. I'm thankful to him and to the ICGEB environment. Actually, in the ICGEB, I learned, and in his lab, I learned to perform different kind of experiments independently. So I'm thankful to Dr. Dinesh, to ICGEB, to the current director, Dr. Salunke sir. So I'm thankful and for all the support, and in just 6 months, I'm getting support from the clinician, from the IS officer, from the commissioners, so many people that they are believing in our company and to the ICGEB environment. So thank you very much for this opportunity. Thank you.
Thanks. Kirti? Okay, I think there is some. Anil Singh.
So thank you for this opportunity. I thank PM sir, Dheeraj and Director sir for organizing the first alumni meet. So I am Dr. Anil Singh. At present, I am working as Principal Scientist at NIPB, National Institute for Plant Biotechnology, Pusa campus. I did my PhD from Pantnagar University and I joined ICGEB as postdoc. Dr. Shiva Reddy is here. So thanks for selecting me, sir, for postdoc position at that time. And after 1.5 years, I moved with Professor Sopori and Dr. Isnaizlo. And whatever I have learned from ICGEB, I am utilizing that till today. So as a lot of people have said, whatever I am because of ICGEB. Whatever little I am because of ICGEB. So thanks to ICGEB and thanks for organizing this.
Thank you. Thanks, Anil. So my lists are over, but I still see a lot of people. Anyone who really want to say immediately in next few minutes can please stand up. Or we move on to the next scheduled thing, that is the panel discussion, which will be. I can see, yes, Agam is there.
Well, I joined ICGEB in 1998 November with Dr. Chetan Chitnis post my PhD from GNU. I was working here as a research scientist in the malaria group. And before coming to the ICGEB, I have never worked with malaria. I was working on a bacterial system. So I started my journey with malaria here and got a very good experience working in an international environment. And after completing my work here in 2003, I moved to NYU Medical Center. There I joined with Victor Nussensweg and again started a new area, that was the liver stage biology. Worked there for almost like 5 years. And then again returned to India, joined National Institute of Immunology next door to this place. And since then I am heading the laboratory which works on the various aspects of liver stage biology, including basic biology, drug discovery and vaccines. Thank you all the people who were around that time. I can see a lot of people, Tatiana and other things, my senior colleagues. We had a very good environment and which nurtured our science to the best possible level. Thank you all.
Thanks. And yes, Mudgil is here. Yes.
Hello.
Rajesh. Okay.
Good evening, everyone. And it's really nice to be here again in ICGEB.
So it's not that very long. Very recently I have moved from ICGEB to a different place, and I started my journey in ICGEB with the bioenergy team, and it was a great privilege to working with Shams, Naseem, Shashi, Pawan and Shirish, all. And then we did very great work and we organized a lot of meetings, and we had a great visibility in the whole campus. And everyone was saying, you have booked all the room every time, don't do it. So this was the great thing to hearing, that it's so much of engagement every time. And very recently I just moved from ICGEB to CII, Confederation Industry of India, where I'm working as a director and leading the medical technology and the life sciences domain area. And it's really good to be here all the time whenever I get an opportunity. And thank you for the invite to have an interaction with all guys. Thanks a lot.
Thanks, Dinesh. Yashwant Budkil.
Namaskar to all of you. First of all, I would like to thank the organizer for organizing this alumni meet. I'm very glad to be back here, and what I want to say has been covered by a lot of people who were speaking before me, and this was the best place and the best time of my life. Dr. M. K. Reddy was my direct mentor, and Professor Sapori was my kind of super mentor. And I had best training and best environment, best friend in my life, and every day was a festival. Like, you come here, you are enjoying your work, you are enjoying your day, going to the canteen, and I'm getting overwhelmed, I'm sorry. So I think if I look back in my life, this was the best time in my life. And the way Punjab previously said, it was an open format. You can go anywhere on any floor to use anything, and your work will never stop. You can work 24 hours, right? You can work 24 hours, there is no hindrance, you can get anything you want. And the way Siddharth said, the best part is you look ICGEB everywhere. I went to US for postdoc, I was still looking for the same kind of format. I went back to India, joined Delhi University, again looking for the same format. And I wish to say this is the best place, one of the best places in India to work and to progress. And I really love this place, and it has a special place in my heart and always will have. Thank you for inviting me.
Thanks. Rajagopal.
My name is Rajagopal. I came here in 1999. Actually, I was a scientist in an ICAR and worked here for 1 year. Then I was offered a job here. So I quit a permanent job in ICAR, pensionable job in ICAR, and took up this job here. I was here for 10 years. I worked with Dr. Raj Bhatnagar for almost 10 years, had the wonderful time, then moved to University of Delhi and been there for the last 13 years. And thank PM and everybody else to have organized this meet, and thank you so much.
Thanks. Dr. Natarajan.
Hi, I'm Natarajan. I was a faculty at the immunology group, what it was called then, but between 1998 to 2010, we worked with Kanuri Rao, Pawan Sharma. Dheeraj was one of the students we had, and this was the first batch whom I taught during the PhD coursework. So had a great time. So between 98 and 2000, we were working on B-cell immunology, B-cell receptor signaling, and then on a cold January morning, Professor Chauhan, the then director, called me and Pawan Sharma to his office. He said that, okay guys, DBT wants us to start a program on tuberculosis. Stop whatever you're doing and get back on getting something to TB. So on January morning, we started sweating because we had no clue what the hell that thing does. Apart from, and then we looked at literature, so we built up something, managed to do something. And in 2010, I moved to University of Delhi's Ambedkar Center for Biomedical Research as a professor, and I'm still continuing, and like someone said, I'm still stuck with TB.
Thank you, thank you. And before we go towards the last panel discussion, I can see Andrew sitting there.
So you can just say a few words.
Actually, I don't qualify as an alumnus. I'm from JNU and I spent 2 years over here from 98 to 2000. But I see from the number of people, including Gayatri and Puran and others who overlapped with us and came over here as students, that maybe I do qualify. ICGEB at the time was housed, the plant molecular biology was housed in the School of Life Sciences before this building came up. So we have an association from the early 90s with ICGEB. But at that time, it was an international institute which only employed postdocs. So I'm not really an alumnus. I was one of the last employees and probably the first one to be sacked, because even though I had my first paper, I went running back to JNU. So my one abiding thought about ICGEB is that coming from the university system, it was an extremely resource-dense and privileged place to do science. And it's something that we should remember. And I went back, honestly, I was here with a bit of guilt, and I went back also with a bit of guilt, because the ease at which one could do science, right from procurement towards running experiments that worked because your buffers were not contaminated, and because when you pressed buttons they worked, was something that we should appreciate. And I hope this institute continues. It has been for the last 25 years since I've been back at JNU. And I'm glad to see so many people from that time that overlapped with me. Thank you, Dheeraj.
Thanks. Thanks. Prakash, why don't you also just quickly introduce?
Thank you. Thank you for organizing this. I am Prakash Mishra, and I will just quickly answer two questions. Why I joined ICGEB and what after ICGEB? I came here after doing my BSc Physics Honors and MSc Biotechnology, Advanced Diploma in Bioinformatics from Pune University. After doing MSc Biotechnology, I wanted to do PhD from ISC, MBU. But by any chance, I was not able to clear that, and I came to ICGEB. And I thought, from being from Bihar, I thought if I will not be able to do a good science, I will clear UPSC. So, but anyway, when I joined ICGEB, during coursework, we had lectures from Dr. Naveen Khanna sir, from Dr. Chetan Chetnes, Dinesh sir, and Dr. Kanori Rao. And then I decided, no UPSC, I'm going to do science. And now, after, I joined Dr. Amit Sarma's lab. I did structural biology and bioinformatics, a PhD in that, in malaria. And then I joined in 2000 at Guru Nanak Dev University, Amritsar. And there, basically, what after ICGEB? Certainly ICGEB was the best place to do PhD, and it's very easy to work over here than to work in Guru Nanak Dev University, which is at a border of India. So, but anyway, whatever culture and whatever things that we learned here, I'm trying to pass it to my students, and I told them just a week back that when we were doing PhD here, we were not having attendance system. So I don't know if it is there now or not, but at ICGEB, at GNU, we have attendance system. So I told them, in spite of not having this attendance of PhD student, we were working like anything here. And in spite of having attendance system at GNU or some other institution, you will find most of the students absent in the lab. So we are just trying to create students having those moral values, those ethics, those scientific temperament, and I think a bit we are successful, because when I joined GNU, when I used to ask them, what do you mean by protein structure? What do you mean by secondary structure of protein? None of the students was able to explain Ramachandran plot. But now when I ask BSc students, when they come to my department for MSc, then I get answer like, what do you mean by secondary structure? What do you mean by Ramachandran plot? So they are being taught, because whatever we taught to our students in MSc, now they are going and becoming faculty in colleges, and they are teaching those BSc students what do you mean by structure of protein and all that, what we are teaching. So I think we are not only doing research, we are also doing some social teaching and all that. Thank you.
Thanks Prakash. Let's have a big round of applause for everyone who just spoke. And moving on to the final session within this auditorium, let me just invite our Director General Lawrence Banks, Dinkar Salunke, Director ICGEB, Sanjay Singh, you may please join us for a quick informal, formal, whichever format you want to keep it. Pawan Malhotra, Shishendu Mukherjee had to leave, so Suresh Nair and Suzanne. So please. So I think a lot of background, about how this meeting was being planned since last so many years has been discussed this morning. So I won't spend much time. There are a lot of ideas which we have been hearing as we were planning this meeting. So it's better we hear from all the people who were involved in this. So we can start with Lawrence.
Okay, so I'd just again like to say what a wonderful day it's been, and thank you so much to everybody who's been able to come. It's really been tremendous. So we've been bouncing a few ideas around, and I think the network that we have of the alumni from ICGEB is absolutely amazing. As we just heard from Suzanne, the numbers are really quite remarkable, and really I have to say we've been slow in getting this activity going, but I think the time is really right now to really build this association. All of you here in the room, but the thousands of others that are all over the world, and so what we intend to do in this little session is just to sort of also look for ideas from you about how we can make this association valuable for you and for what you do. Maybe you can also in return help us with, when we put you on our mailing list, you can help cascade information about ICGEB to your own networks. That is something that's, even now, after 30 odd years of going, we still go to many countries and meet people and they've never heard of us. So really this sort of spreading the word and what we do is really very, very important. And really something that I think we really want to do is to use this initiative, which again I have to say a massive thank you to the team here in New Delhi, which they did this at remarkably short notice. But we're going to make this a regular event, and we're planning to do another meeting next year in December. I believe the dates are 6th to the 8th in 2023. We'll have more time to get more alumni and it'll be an even bigger event, so I really hope that many of you will also be able to make that. And of course, we're always looking for assistance and help to expand what we can do. And so if any of you come across any schemes or activities which might be of benefit to the ICGEB, then please do let us know. So thank you very much indeed.
So maybe, yeah, I think Sanjay Singh is.
So thank you. First is this so important day that came back to the same place where I have worked for so many years, got so many ideas. I feel that we should keep revisiting the mantle of ICGEB for why ICGEB was built. And what I read when I came to ICGEB or coming before to ICGEB, ICGEB main role was educating biotechnology in LMICs in most of the countries, in biology, in bioinformatics, in plant sciences. But it was also, mantle was that it should be very transnational. It should also create products and or train people in those countries, either in Bangladesh or in South Africa or in Kenya or in Indonesia or in Cambodia or in Colombia. I think we should somehow, because there are a lot of universities, there are a lot of systems which produce PhDs and which teaches everything. So ICGEB must do it, but under a particular segment, it should not be only it should be institute with a difference. And that is my thought process. And anything which I can do into this, I have talked to you, I have invited you, come and visit Pune, what we are doing. I will be very happy to participate. Thank you.
Thanks, Dr. Sanjay Singh. So, Dr. Salunke?
I think we started this meeting today with talk by Dr. Sanjay Singh, and he gave quite a lot of messages. And simply his messages, if we can fulfill, we would have actually fulfilled the purpose for which we set out to meet. As in the morning, Dheeraj said, we have from youth, rather early days to adulthood, in the sense we have reached about 35 years of age. In that sense, we are a mature institute. And as I can see now, not only Susan's data, but listening to the profiles of people who have done their PhDs and postdoctoral fellowships. So in that sense, you qualify to be alumnus of this institute. So you cannot run away, Andrew. So, which means you also need to contribute to make this institution to be the way you imagine it should sustain and continue. So I think Sanjay said it practically openly, that the ICGEB was conceived to be substantially self-sufficient, and we all together can make it that way. And I think this is first step to reach out to the alumni, and we should come up with the ideas in terms of how to make ICGEB of what vision that was presented by Sanjay for ICGEB. And I'm glad that he has promised to be engaged with us for a long time to come. And I'm sure many of you, considering the kind of emotions you expressed, you will be associated with us in actually making those dreams very successful. And we promise you that ICGEB will be institute of your dream. Thank you.
Thanks, thanks. Maybe we go to Suresh.
This has been a very, very long journey. It's over 30 years. And it's a pleasure this morning to have caught up with several of the people I've been associated with one way or the other. As a lot of people said, that we had a very open lab system at ICGEB. On paper, you were A student or B student. Every student on ICGEB was your student at the end of the day. If they had a problem which was associated with something that you worked with, they approached you, and it was your duty to solve that problem. And as someone already remarked, that if you couldn't solve it, they'd be sure that they'll be there the next day to ensure that you solved it for them. That said, over the years, ICGEB has evolved in different ways. Programs start getting changed, new programs come in. To be, as we just heard on the panel, that ICGEB had to be innovative. To be innovative, you need to have new ideas, new programs. So a set of programs was set up 30 years ago. Those programs have changed slowly as part of the evolution. But what we need to do is speed up these changes that we need to do at ICGEB, because we have to make ourselves relevant. That said, how do we do this? So as Sanjay has already mentioned, that he'd be more than happy to be associated with ICGEB. We also need to have a program with the panel of scientific advisors that ICGEB has, to have an interactive session to evolve new agenda. So one of the agenda, the top arching agenda, was to have the kind of agenda that we had started this ICGEB system 30 years ago or 35 years ago, but we need to evolve new areas of work. Why? Because if you have to make ICGEB relevant, you have to ensure that the common problems or the problems that are being faced by the world are addressed. And this is extremely important, because students look up, or researchers look up to ICGEB for a direction, for interaction. So we have to innovate. And the faster we think in these lines to innovate and start new programs is something that ICGEB needs to do much more rapidly than we were doing now. So one of the things was to get in touch or have an interactive session with our panel of scientific advisors. The other thing is to have alumni, or the alumni who are working in different areas. So it's a good thing to have the email addresses and contacts put in there. It's a good thing to also have the background as to what work they do. Because if I need to do something that they are a specialist in now, I know who to get in touch with. So this is a wonderful initiative. We were talking about this. I remember about 3 years ago, Dinkar emailed me and said, can you send me a list of students who you know, because you're one of the oldest guys here and you must have interacted with a lot of people. So I did send him a long list, and he said, thanks a lot, and we'll get to hear from you. I would get to hear from him very soon. But that didn't happen for over the 2.5 years, and I can understand why. But I'm glad that it did happen. I'm so happy to have met with many people I was associated with day in and day out. And I've superannuated, and I'll be here for a few more months. But seeing this place prosper would give me a lot of energy that I can sustain myself for the remaining part of my life. Thank you.
Thanks, Suresh. PM. Thanks, Susan. PM.
That's a wonderful place. When I moved from US to ICGEB, in fact, I found better than many other US lab, and I never had to look back when I joined ICGEB. And I personally feel we grew together with you. So we will require your suggestion that how should we make this alumni more better and better, and how should we contribute so that ICGB can reach to some of the heights, and so that we can also convey, as our DG said, to other countries who are not part of ICGB, that be part of ICGB. We build up science, we develop science together, and do some of the translation research, which Sanjay has said, for the community and for the society. So I think we need your participation in this alumni, organizing this alumni next year, as our DG said, in a big way, so that we can again, we might have missed few people in our list, so we want your help so that they can join in those links which we have, Dinesh and Susan has them. Please enter, tell your friends that we might have missed them, but please do come into this and join in the links, and also please suggest what we can do in this direction, which has been suggested by many of our panelists. Thank you very much again for coming to this place.
Thanks PM. As we head towards the end of the official part of this meeting, I'll just add to what Pawan just said. For organizing the meeting next year, which Lawrence also spoke earlier, there is a plan that we have alumni not only from Delhi component. I know we had Stefano today, but there is a plan to have alumni from Cape Town as well as Trieste, to make it much more vibrant, the way ICGB is known for, representing multiple countries and nations and cultures. So we will need volunteers. I know Das had already volunteered that he would like to be involved more with organizing this. So I'm sure, I know if I ask, I will always get affirmative response. So in terms of help in organizing the next meeting, a little bit more detail of that will become visible on the websites, and through emails will come to you guys soon. The dates are for the timing, I think 6th, 7th, 8th, that Lawrence just said, is mostly fixed. So coming to the final part, I need to thank everyone. I think
most important thing we need from all of those who are here is to fill in those missing dots.
Yes.
That is, I think, connect us to rest of the people who are not in our list.
Right. So Dinesh has made a link on the website, which allows you to input details where you are, your current affiliations, email, phone number, recent photographs, so that if you have changed a lot, we can still recognize. So. Right. So a lot of ex-students have really entered their detail, and that list I saw today has gone beyond 50 or 60, but there are many more who are left out. So my request is that check at that link first, and then circulate among people and with whom you are in direct contact still now. So that will kind of populate the list automatically. And the plan is that once we have sufficient number of people, will have a kind of alumni booklet which can be in a virtual format or physical format, doesn't matter, but that will be ready, can be made from that. So is there anything else that I'm missing and anyone wants to quickly add? Okay, so the floor is open for suggestions from the audience side. Or participants, I should say, not audience. I know we all are tried.
I think the one new area that needs to be pursued is the emerging pathogens, and that may include the viral pathogen as well as the bacterial pathogen, protozoans which are still emerging, not a very prominent problem for the developing countries, but in coming future they will be a real problem. So one of the examples I can say, like Babesia. Recently we had the problem with the corona. Now monkeypox. Now some more viruses are bound to come. So we have to think about how to deal with the emerging pathogen and probably start a division which works on the emerging pathogens and try to develop technology which will be in parallel to their development, or we will be ready ahead of time when the outbreak comes. So that is where one area which I just thought could be thought about or introduced, other than what is going on, already going on.
Yeah. Thanks, Agam. Richard.
I think coming from plant science, I want ICGEB not to lose focus from, remove the focus from plants to all other systems.
That's not going to happen.
Plants need to be as vibrant, as financially supported as they had been in the past. That floor is our pride. You know, anywhere you say that you're from Plant Molecular Biology Lab, from ICGEB, people pause and look at you. That needs to be sustained. How we go about it, we can plan. Yes.
Shweta.
Yeah, I'm coming back to how to generate the alumni this thing, and I think so. I'm very thankful that you guys gave us these sweatshirts with zipper on, and I think you should start selling it on the online so that wherever we are, we can buy more and wear them and advertise freely for you. So
it's an excellent idea. Yeah. Okay.
Actually, now having been an alumnus and now a little aging scientist, many of these networks are sustained with two aspects of funding. The funding of students' existing mobility to alumni groups that are established, and also the reverse funding for alumni to spend some time in residence at their alma mater. So this, I think you should look at in terms of a people-people bonding, because it's not so much of resources and science that will happen, but the people that, at least the PhD students who spend 5 years together, form bonds which are irreplaceable, and the short-term science that scientists like us who spend time here, also formed interactions. And being able to translate that towards supporting existing students is something that needs support from the funding pattern that you have. Thank you.
Okay. Hop. Okay. Yeah.
Okay, thank you. That I took very some small thing, because now you as Dr. Narat said that okay, ICGEB is making innovation, but I think that now is a more program is more competence. The thing is that from our country's story, because I come to be that Vietnam that from when I went to Japan in the late 80 years, I was told by some Japanese professors that okay, Japanese government can support Vietnam in four topic about biotechnology, informatics, new material, or yes, and biotechnology. But whenever Vietnam became an official member of ASEAN country, so ASEAN countries, Vietnam is a member since about 1995, right? But 1995, that's when America's lifts the embargo to Vietnam. So see, that's from that time, Vietnamese young generation can go to such as ICGEB or go to some developed country. So still that means still gap about the technology between Vietnam and the rest of the world. Even in ASEAN country, it's a big gap now between Vietnam and Thailand also. Okay, so the things how to say that makes more competent. Because now the ICGEB, when I come to here almost second or third batch, because of the first batch go to Trieste, only one or two or few person, and then second batch come here by Dr. Suresh Naino as Dr. Lan Wang or Dr. Tu, okay, and then I came with Dr. Gia and then Ho also. So, okay, so but now how to say that? Because now in Vietnam, ICGEB is a very good reputation. Because when you came back, yeah, I fulfilled our job. It's okay. So that you recognize that okay, ICGEB is very good scientist, very thoughts about technology and knowledge. And of course, the other by human being hospitality from the Indian people. So that's the reason why it's a good reputation in the academic session. But now the situation different because the other young generation. So then, of course, now situation before we only few person. Normally, the older person went to the socialist country, but now, but at that moment, at the 90s, 95, we went here. It's a very, very good job. Yeah, but now from the 2005 up to now, so it's more open in Vietnam. So this young generation say. Of course, I think that we would like to introduce a very good and young generation come here to get about a good training about master or PhD or postdoc. But of course, now young generation have a more chance. If they very good, they would like to go to America or go to the other. So of course, still Vietnam have many good something, but they cannot go by a long time, such as a PhD for 3 or 4 years. So if you make competence by about how to say about the sandwich program. So in this case, there are two things. The first, whenever we discuss in the Vietnam topic, we can show the common interest. So it's a more practical in about the developing country. So that's mean about the problem in developing country and about academic solution from here. Yeah, it combined together. So that means that in this case maybe we can thinking about the co-funding. Co-funding is not only from the ICGEB but also the matching fund from the about Vietnam or the other developing country. So that's a thing that because if we focus only about the young generation, very good at it, because we did a lot of the recommendation, but they got fellowships, they went America or go to European country or go to Japan or whatever. So I think that here, whenever we have a good scientist and a good facility, but we need about the rise source of PhD student by sandwich because they got married or they may have some reason they cannot live family for long time. So maybe once Switzerland come to work together. Okay, that's my small thing. Thank you.
Good point. So the good reputation of ICGEB is because you guys went back from here, and it all credit to you guys. So it's time to thank all the panelists sitting here. Director General Professor Lawrence Banks, Director ICGEB New Delhi, Dinkar Salunke, Dr. Sanjay Singh, Parmalhotra, Suresh Nair, Suzanne, all the people who participated in discussions, talks, questions, answers, and having coffee together outside. Thanks to everyone. This meeting was, done on a very short notice, which has been coming out repeatedly from all of us, that we had a very short time. So it required a lot of support from people within ICGEB, and I must say thanks to Dinkar Salunke, Director ICGEB, who really ensured that we had everything at our disposal to ensure that things move. And Mr. Matthew is the Administrative Officer AO at ICGEB. The team of, I think, me and PM working together, I can kind of take credit that the team really worked well, and we managed to handle a lot of things at the very last moment. The administrative support, I have to mention name of Shweta Gupta, and from administration, right from I think third or fourth discussions when the meeting actually got kicked on paper. She has been with us, and I think all of you who are here have received multiple emails from her, for every finest of details, whatever was possible. So she kept everyone in the loop, and hats off to her that she managed this really, micromanage this whole organization. So a big thanks to Shweta. And yes, I was coming to that. And behind the scene, yes, Dinesh really, helped. And one of the, visible part of that was how he developed that website, which allowed a lot of people to connect and, populate the list of alumni. And finally, the people who have come, the alums, and without whom this meeting is nothing. So a big thanks to all of you who took out time and came back to ICGEB. So thank you all. I think we are really tired now for listening multiple talks and discussions. So I think it's time to get a little spiritual, move out, and have some energy. So thanks once again.